And I would like to start this session with a general overview regarding the microbiota, as this is the first lecture, and then have a brief introduction to the gut microbiome and current knowledge regarding the cholestatic liver disease. So, in general, human microbiota is 14 square microbes, which is located mostly in the gut, but you can find some microbes almost everywhere as the studies proceed regarding the sequencing of the material of skin or in saliva. But so far, current knowledge is most extended regarding the microbiota composition in the gut. As most knowledge is gathered from bacterial composition, we currently know around 1,000 mostly anaerobic bacterial species in the gut. So there are mostly firmicutes and bacteroidetes. It is like if you want to see how severe the dysbiosis is, the ratio of those two phyla is quite important. But we have to not forget that the gut microbiota is definitely not only bacteria, but also other organisms like viruses or even fungi. And the composition is, of course, influenced by environmental factors. I think one of the most important when speaking about the gut microbiota is the role of the diet, but of course the oxygen and the temperature. As we will be talking a lot about the genome sequencing in gut microbiota, you can see that in our body most of the genes belong to bacteria. So let's just put a question if our body really belongs to us or to the microbes. But it is important to say that the gut microbiota composition is formed in the first 3 years during childhood and is essential for the immune system maturation. So as you can see, there is quite a progress in knowledge in the microbiota. This is mostly caused by the next generation sequencing techniques, starting around 2010, and really you can see hundreds and hundreds of articles on PubMed regarding the microbiota. As it is getting quite an attractive topic, not only in the gastroenterology but in other branches of the medicine. So we have the data on the specific microbiota composition in various diseases, including obesity, diabetes, but even the diagnosis like schizophrenia or autism. Of course, there is a vast knowledge regarding the role of the microbes and their composition in inflammatory bowel disease, but evidence is emerging in specific microbiota composition and its role in the liver diseases. So why should we even bother looking in the microbiota in the liver diseases? We have to realize that the microbiota, the microbes themselves and their metabolites and antigens are streaming through the portal blood and leading to the activation of various receptors on the liver cells, including the copper and stellate cells via pattern recognizing receptors. The most evidence is on the toll-like receptors and non-like receptors, leading then to recruitment of the immune effector cells and upstream regulation of the costimulatory molecules on antigen-presenting cells, leading then to the antigen enhancement of the antigen presentation T cells and eventually to increased biosynthesis of chemokines and other signaling mediators in the liver. But we also have to realize that most probably at the beginning of the whole process, the disruption of the gut barrier function is at place. So, for example, in case of inflammatory bowel disease with the concomitant primary sclerosing cholangitis, we really need to stress out the question if the gut disease or liver disease come first. But we suppose that it is quite deeply interconnected. And looking at the serial markers of the gut barrier function could be essential for the future studies as well. I suppose that, that the field that is not so well understood regarding the microbiota and liver disease is the liver transplantation, or even the solid organ transplantation in general. Because of course there are various environmental factors, including the antibiotics, including of course immunosuppressant, including the altered anatomy post transplant, and all these factors could lead to alteration of the microbiota. But we really need to look into it if this could be somehow used as a biomarker for the complications like acute cellular rejection or other complications of the liver transplantation, and stratify the patients into the risk groups for the prognosis of the liver graft. So the cholestatic liver diseases in general, there are several drug-induced genetic IgG4 associated cholangitis, but we will be talking, of course, mostly about primary biliary cholangitis, PBC, and sclerosing cholangitis, PSC, because the knowledge regarding the other diseases is not that well known, and of course you have what used to be called overlap syndromes of those diseases, for example, with autoimmune hepatitis. So briefly about primary biliary cholangitis. Recently it was renamed because it somehow stigmatized the patients, which were called like the primary biliary cirrhosis, but this wasn't quite proper. We can oppose to primary sclerosing cholangitis say for sure that this is the autoimmune liver disease involving small bile ducts and leading to ductopenia and subsequent damage with the bile acids. That's the reason why UDCA is a first-line treatment in this disease. You can see even an old picture with a typical face in a woman patient. Recently, there was an article because in the PBC there is not much known about the role of microbiota, but there was an article with a three-phase study with a validation cohort from Tank from last year, and it was possible to see the reduced species richness in the PBC patients as compared to healthy controls, and the distinct pattern of the microbiota composition in UDCA naive patients, which is quite interesting because there was a subsequent cohort with the patients who were treated with UDCA for 6 months, and this led to dysbiosis improvement, and also it was described in the study that the innovation of epithelial cells was highly correlated with the abundance of Enterobacteria say. So this could really stratify the patients into the risk groups, and to see how severe could the progression be. So as for the primary sclerosing cholangitis, of course, it is still a liver disorder of unknown etiology, characterized by inflammation, fibrosis, and the stenosis of the biliary tree, both extra and intrahepatical, as you can see the typical bleeding and the cholangiogram, leading eventually to cirrhosis, liver failure, transplantation, or death. Liver transplantation, of course, is the only currently known treatment of the disease, even though the disease recurs in up to 44% of the patients. And it is important to say that in the northern Europe it is the most common indication for liver transplantation, not so in the southern part of Europe, but still it is the leading indication in Europe. As to understand the role of the microbiota, because I mentioned the role of microbiota in IBD previously, we have to say that up to 80% of PSC patients also have concomitant inflammatory bowel disease, which is considered to be a phenotype distinct from, let's say, ordinary ulcerative colitis and Crohn's disease. It is clinically characterized by a mild course pancreatitis, red-sided predominance of both inflammation and colorectal neoplasia, and sometimes with rectal sparing and backwash ileitis. As I mentioned, high neoplastic potential potential involves not only the colon, but of course there is a severe risk of cholangiocarcinoma. So for both neoplasia, the patients should be properly screened. So therefore, as recommended by ECHO, they should have the regular colonoscopy at least during annually. So microbiota in PSC, we have to again focus on the gut barrier dysfunction leading to exposure of microbes to the liver, and the presentations leading possibly to the inflammation and triggering the autoimmunity processes. But of course, as the cholestasis emerges, also the composition of bile acids is quite different, and there's like a vicious circle, because the altered composition of bile acids, of course, influences the composition of microbiota, and the same thing is clockwise. So we were thinking that because of the pathogenesis that we already know, we hypothesized that in PSC-like models of rats, there were bacterial overgrowth. They developed something resembling PSC, and there has previously been a successful antibiotic treatment in PSC. So therefore, we thought that there should be a crucial role of microbiota in PSC pathogenesis. So it is my great pleasure that we could present some data that we gathered through last year with our cross-sectional single-center study of 106 individuals with PSC patients with or without inflammatory bowel disease. We obtained the stool samples at a regular checkup, and performed a state-of-the-art Illumina MySeq sequencing of the 16S gene, which is quite specific for bacteria. These are the variable areas, and which are really specific for each bacteria. So you can go into very low levels of the taxonomy to really see what's in there. What we saw that all the diseased patients with either liver or gut disease had a lower microbial diversity, but this well correlated with the disease activity. As I mentioned earlier, the patients with PSC IBD have mostly mild pancreatitis, while the UC patients have usually more severe inflammation. So we saw the lowest diversity in UC controls, but it was of course also in PSC patients with just liver disease. So we can conclude that the presence of ulcerative colitis in PSC patients is associated with a reduced diversity. Very similarly, this works for the beta diversity, which we see on the plots in here, as the global composition really distinguished the liver disease from the healthy controls, and also each subphenotype of inflammatory bowel disease, the ulcerative colitis on one hand and the PSC IBD on the other hand. So really, this is just for the illustration that there are all the patients in these plots, and you can go very deeply throughout the taxonomy, and we saw significant differences at each taxonomic level. But we thought that the most interesting would be to go really down. So we focused on genera and species. And this would go further down to the species level. I cannot discuss all the bacteria that we found more or less abundant in each diagnosis, but just what seemed interesting to us, that for example, Faecalibacterium prausnitzii, which was proposed to be a really potent probiotic, was significantly decreased not only in ulcerative colitis patients as described in the previous studies, but in the PSC cohort as well. And this is interesting because each study regarding the microbiota composition had quite different methods, but still we were able to duplicate some results regarding the Veillonella, regarding the Streptococcus, and what was the new finding was the high, very specific abundance of Rothia in PSC patients, which was interestingly independent from inflammatory bowel disease. So from this we could conclude that the dysbiosis in the gut is driven by the liver disease, not the inflammation of the gut in this very complex PSC IBD disease, which could be quite interesting. Recently, and interestingly, Iwasawa, there is a Japan study, reported that there was a significant decrease of Rothia and Haemophilus in saliva in PSC patients. So we, on one hand, have a decrease of those genera in saliva, but on the other hand, it goes up in the gut. So we can conclude that in the advanced liver disease, there is a translocation of oral microbiota to the gut, but we have to address why this is happening, because nobody knows this so far. So what is also interesting that in our multivariate analysis, as we put all the metadata, we were able to see that regardless of concomitant inflammatory bowel disease, presence of overlap syndrome of autoimmune hepatitis with PSC had a specific bacterial pattern. So as you know, to really confirm the overlap syndrome, it is sometimes necessary to perform the liver biopsy. So this therefore could somehow lead to for search of the biomarkers to really distinguish phenotypes of the liver disease. What is interesting that of course we are now thinking, now that we know what kind of composition is associated with each disease, we are thinking about the modulation of the microbiota, which could be done either by probiotics and prebiotics. FMT will be addressed in the lecture by Tibor after me, and of course antibiotics. In PSC, the current knowledge is mostly focused on vancomycin. There was a meta-analysis by Daman recently, which included almost 100 patients, and the article itself, it is called "Evidence that the vancomycin is an alternative treatment for PSC." And the reason why is that we suppress specific bacteria in the gut, leading to the improvement of the structural changes in the biliary tree. So therefore, antibiotics in general or modulation of microbiota could be promising. Ladies and gentlemen, from this brief message, let me please conclude that most likely microbiota plays a crucial role in the pathogenesis of various diseases, but definitely in the cholestatic liver diseases, and the most evidence is for primary sclerosing cholangitis. Different diseases are characterized by specific gut microbial composition, as I demonstrated earlier. So this could lead to finding a potential biomarkers in diagnosis of the disease phenotype to predict the risk of associated complications, for example, the PSC and IBD. But of course, there is a egg-chicken principle once again, and to see the prognosis either in the primary disease or even in the prognosis in the liver graft in patients after the liver transplantation. So therefore, this could possibly lead to replacement of invasive or more expensive methods, because we could recruit the biomarkers either from this tool to really see each microbes, or to search for the antibodies against the specific microbes in the patient's blood. And hopefully the microbiota modulation could be a promising target for treatment of both primary and recurring liver disease, but it's of course has to be addressed in future prospective studies. So I would like to once again kindly thank the organizers for the invitation, and I'm ready for our questions. Thank you.
So thank you very much for your comprehensive overview and update of this hot topic. So we'll open the discussion right now, and I would like to ask the audience to raise your questions. One or two questions from the audience.
Yeah, thank you very much for your very interesting lecture. And would like to ask you, what kind of index of bacterial diversity was used in patients with primary sclerosing cholangitis?
Actually, as for the alpha diversity, all of them, there was Shannon, there was the species richness, so it addressed both richness and the evenness of the bacteria, and in all of that we could see the same. So there were like 3 indexes for alpha diversity, and we could duplicate the results in each of them.
Thank you.
So I also would like to ask you one question. So you have mentioned at one of your first slides that there are archaea, fungi, and viruses present in the gut lumen. Do we know anything about these, let's say, different microbiota and their possible role in the pathogenesis of PSC or PBC or other liver or gut diseases?
Thank you very much for this question, because it is quite a hot topic. There is evidence of the presence of fungi and also the interkingdom interconnection between bacteria and fungi in inflammatory bowel disease. But in the liver disease, so far, nothing is known. But I could maybe reveal something from our kitchen, that we really wanted to repeat this experiment that I showed also for fungi, and there are some promising results also specific for the fungal dysbiosis in the same data set as I showed for the bacteria. And to obtain the knowledge regarding the virome, it is much more complicated from the technical point of view. We have, of course, DNA and RNA viruses, so this works quite a different way, but the protocols are emerging that it is also possible to have a description of rough description of virome in the stool as well.
Okay, thank you very much.